All right, so welcome back. I don't know, did you guys figure out the answer to rigor mortis? Kind of cool. All right, I'll help you understand like how muscles function and stuff. And it is one of those things that could possibly go wrong. And when we talked about it, I also kind of want you to think. And I know these are a little bit ridiculous examples, but when you think about like zombies and the way that they function, right? I mean, right when they move, it's real like lurchy kind of motion. So think about like how what's going on with the ATP, the myosin head, and all of that, the electrical signals. So if that's how a muscle contracts, and we'll talk a little bit more about how they contract now too, think about what's going on in a zombie and like what do they look like and what might be the cause of like a lurching kind of action versus a smooth kind of action. Okay, it's a good thought exercise. Okay. All right. So anyway, so let's just talk about some of the structural proteins. So these are the proteins that are keeping the sarcomeres together and making sure that, I mean, think about it. You have to make sure that those actin and myosin heads or the actin filaments, the myosin heads are nicely aligned so they can move past one another. Okay. So these, again, so these structural proteins, they'll keep these things aligned. They'll also give your muscles its elasticity, so you can stretch muscle to a point, without tearing it. And then it'll also spring back on its own. Okay, I mean not quite with the same force as a rubber band, but it will go back. It's not like a piece of gum. Okay, and these structural proteins will also link those myofibrils, so the actin, the myosin, to the actual sarcolemma, to the cell membrane, and then to the extracellular matrix, so to the other connective tissue, to the tendon. Yeah, I'll just hide me here. So one of the first ones that we're going to talk about is this protein called titin. It's called titin because it's really big. So it's a fairly thick filament. You can see it right here. It kind of looks almost like a spring. Okay, so it goes all the way from this M line. Remember that M line is going to be a supporting area, kind of helping support that central part of the myosin. Anyway, so it's going to anchor from that M line all the way to the Z disc. Okay, and as kind of they have it, it's sort of like this in real life too, but as they have it, again, it looks like a spring, which means it can stretch quite a bit, about 4 times its resting length, and spring back unharmed. Again, this is what's going to help your muscles recover from being stretched. And again, within reason. Obviously, if you overstretch your muscles, yes, they will rip. Okay. That M line is made up of a protein called myomesin. It connects to that titin. You can see here again, it helps support that central part, that sarcomere. You won't see it in this image, but there's another one called nebulin. It's a protein that wraps around the thin filaments, and it's one of the things that's going to help them align with the thick filaments, and also it'll anchor the thin filaments to the actual Z disc. And then finally, another protein, and you probably have heard of this, dystrophin, will help link those thin filaments to the actual cell membrane and transmit that tension to the tendon. Okay, so this is kind of a key concept here. Have you guys heard of muscular dystrophy? Probably have, right? People with muscular dystrophy, right? Their muscles don't function anymore. They're very weak. These people are usually in wheelchairs and stuff like that. What happens with muscular dystrophy is this dystrophin, this little protein, is not made properly. So basically, by this not being made properly, those thin filaments are not linking to that cell membrane. And since they're not linking to that cell membrane, all of that tension is not being transmitted to the tendon. So what ends up happening is that since we're not transmitting that tension, it's all being contained in that sarcolemma in the cell membrane, and so the cell membrane of muscle cells will start to rip. And if you start ripping putting big old holes in cell membranes, the cells will die, and so basically the muscle wastes away. And that's basically what muscular dystrophy is. Okay, it's a problem with this protein called dystrophin. Okay. All this curve is trying to say is that there is a optimal overlap between the myosin here in pink and the actin here in blue. So at this sort of overlap is where you can generate the most force, the most tension. If they are overlapped completely, well, you cannot produce any more force. That is all that you can produce because they can't go anywhere. Likewise, if the muscle is stretched too far, you don't have enough of those myosin heads overlapping with the actin filaments. So again, you cannot generate a very strong force. And then obviously, if there's no overlap, you cannot produce any force. So if you stretch the muscle way out, that's it. You can't produce any force is basically what it's saying. Versus if the muscle's already completely shortened, well, it's got nowhere to go. Okay. All right. So how do we get muscle fibers, how does the muscle fiber actually get that signal to contract? So let's bring in that neural part of it. Okay, I mentioned this to you guys before. So where the neuron and the muscle meet, we call that a neuromuscular junction, or it's also known as a synapse. Either term is fine. Okay, ever so often you might see it written the other way. Instead of neuromuscular junction, you might see it called a myoneural junction. Some textbooks and some sources use that term. Same thing as neuromuscular junction. Okay. Basically, so here you can see here's a motor neuron. The end of one neuron splits. Okay, so the end of the axon will split, and so you can see here's this branch, and it's going to end in what are known as synaptic end bulbs. Okay, and so you can kind of see these bulbs a little bit better. So again, here's one neuron, just one nerve cell coming down, and then its end splits into these different branches, and then it's those different branches that will go to different muscle cells. So this long thing is one muscle cell, this is a muscle cell, this is a muscle cell. Okay, where they come together, again, neuromuscular junction. Now, this is a key concept. I don't know how well you'll see it here. You can kind of maybe see it here. There's like a little space. Can you see there's like a tiny little gap between the two? Interestingly, they don't actually touch. It's almost like that kind of thing that you would have done with like a sibling to annoy him. You know, I'm not touching you, I'm not touching you, that kind of thing. Super close, but you're not touching the person. But the effect, you might as well be, because it's just that annoying, right? It's sort of the same thing. So these neurons are not actually in, there's a tiny, 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 tiny gap between the neuron and the actual muscle. Okay, so that space, let's see, where's my little pointer? Did I lose my pointer? Where'd you go? That little space is called a synaptic cleft, or a gap, or a synaptic gap. Okay, hopefully that makes sense. Let's see, other terms. Where that neuron is going to touch the muscle, it's not random. So it's actually touching it at a very specific location. That location will have lots of receptors for a neurotransmitter. Okay. And that neurotransmitter is what's going to allow that nerve signal to go from the nerve, jump across the gap, and then onto that muscle. So again, that part of the muscle isn't random. It's very specialized. It has lots of receptors for this specific type of neurotransmitter. Anyway, that specific area on a muscle cell is called its motor end plate. Okay, so again, that axon branches. It ends in what's known as a synaptic end bulb. Those synaptic end bulbs are not in direct contact, but in very close contact with the muscle cell, and that special area of the muscle cell, special area of the sarcolemma, the cell membrane, is called the motor end plate. Okay, so this is located on the muscle cell. And there's a small gap, either called the synaptic cleft, synaptic gap, or you can just call it the gap. Okay. So here we see this sort of zoomed in. So again, here's the axon, the synaptic end bulbs. So here's a synaptic end bulb, and you see these neurotransmitters being released into that synaptic cleft, that space, right? And on the other side is that motor end plate. So here, let's just zoom in one more time. So here's that synaptic end bulb. Here are the neurotransmitters being released across that synaptic cleft. And then here are the receptors that are going to bind to it. Okay. The neurotransmitter that we're talking about is called acetylcholine. Okay, it's acetylcholine. That is a neurotransmitter. So basically, that nerve signal comes down. It can't. If there's a space, it can't. It stops. Right. So it can't go any further. So the way you get that message from that nerve to the muscle is you release these neurotransmitters. Those will go across that little space. They'll bind to the receptors. And since they bound to that receptor, it causes a channel to open. And as soon as that channel opens, you can see sodium goes into the cell. And as soon as that happens, that signal continues down the muscle fiber. Okay, I'll say that one more time so you can kind of take notes on this. Right? Nerve signal will go down the length of the axon. It gets that synaptic end bulb. It cannot cross that synaptic cleft. So what the neuron does is it releases a neurotransmitter. In the case of muscles, that neurotransmitter is acetylcholine. Okay? That acetylcholine will travel across that synaptic cleft. It'll travel across that synaptic cleft. It binds to receptors on the motor end plate on the muscle. By binding to those receptors, channels are opened. Sodium can come into the muscle cell, and that is what continues that muscle action potential. So that's what continues the signal down the length of the muscle. And that's what will eventually go down those transverse tubules and cause the release of calcium, etc., etc. Okay, let's take another look at this. Here's sort of a summary table. So here's a nerve impulse releasing acetylcholine, goes across the synaptic cleft, binds to receptors on the motor end plate, opening up sodium channels. That nerve impulse goes down the length of the cell membrane, goes down those transverse tubules. By going down those transverse tubules, it'll cause the opening of calcium channels in that sarcoplasmic reticulum. So calcium will leave the sarcoplasmic reticulum. The calcium then binds to the troponin on the thin filament, causing it to change its shape. It pulls on the tropomyosin, which then causes those actin binding sites to be exposed. That means those myosin heads can now bind to it. It undergoes that entire process that we talked about in the previous video. And then basically, if there's no more nerve signaling, the calcium is actively transported or pumped back into that sarcoplasmic reticulum. Okay? Since there's not so much calcium floating around in the sarcoplasm, the troponin goes back to its original shape, which relaxes that, kind of puts that tropomyosin back over the actin binding sites, and basically the myosin heads can no longer attach, so the muscle relaxes. Okay, hopefully that makes sense. Please go over this slide a few times. Make sure that you're okay with all of these different steps. Make sure that they make sense to you. In this part right in here, the power stroke, we had talked about that in the previous video, so excuse me, add that back into this story. Okay, so you can kind of add to the story. And then basically it's going to be up to your brain and different parts of your brain, and again we'll talk about that for the next exam. The brain is what's going to send the signals down to the muscles to have that coordinated action that allows you to judge the distance to that cup that's sitting on your desk. That's going to allow you to judge the amount of force that is necessary for you to grab the cup. That's going to allow you to judge the distance from once you grab that cup to your face to where your mouth roughly is, so that you're, when you take that drink, it's a nice smooth thing. You don't dribble it all over yourself or pour it into your lap. That's all the brain sending very specific nerve signals down to your muscles. But basically at the small molecular level, this is what's happening that actually allows the muscle to contract. Okay? That's actually producing this force. All right. So here I just have it written out. Again, it's a slightly different way. The last sort of thing that I didn't mention in that last slide is there is an enzyme called, it's an enzyme. It ends in an ACE. It's going to break down the thing that comes before the ACE. So acetylcholine esterase breaks down acetylcholine, right? And if you haven't figured this out yet, ACH is simply the abbreviation for acetylcholine. Okay. So basically acetylcholine esterase breaks down the acetylcholine attached to the receptors on that motor end plate, so the muscle action potential will cease and the muscle cell will relax. Okay. So that's going to be really important. All right, so let's just kind of talk a little bit about the pharmacology here in the neuromuscular junction. So you guys have probably heard of botulinum, right? Botulism, for botulism, dented cans. Don't eat fatted dented cans because you might have the botulinum toxin, right? I've heard of Botox. Okay, basically Botox is modified botulinum toxin. Okay, it's basically a very weak form of it. Right? So what that toxin does is it actually blocks the release of acetylcholine at the neuromuscular junction. So if you block the acetylcholine, that nerve signal does not get to the actual muscle. The nerve signal gets to the very end of that nerve, it can't cross that synaptic cleft, right? So the muscle never fires. That's how Botox works. So when you inject it in your face, basically the muscle is paralyzed because you're sending all the nerve signals, but the muscle itself, cannot contract. And then where it gets deadly is that, remember, the diaphragm is a skeletal muscle. Okay, it's not autorhythmic like your heart. So basically what happens is if you have this full strength botulinum toxin, what happens is the nerve going down to your diaphragm can't release its acetylcholine. The diaphragm can't contract. You can't breathe. That's it. Okay? You can't breathe, you suffocate, you die. And people have died of that. There have been cases where people get sort of, I don't know, what was it called? Like bootleg Botox, right? They get Botox from a non-reputable source, right? They inject it, they start not feeling well. And what ends up happening is they suffocate, right? And because paralysis comes on fairly fast. You start not feeling well, and the next thing you know, you're paralyzed. And as soon as you're paralyzed, you can't scream, you can't move, you can't do anything. Okay, so it's very dangerous. Curare is plant poison, and I shouldn't say it's from poison arrows. It's used in poison arrows. Okay, so it's used in poison arrows. This works, so basically it also causes paralysis, but instead of blocking the release of acetylcholine, it blocks the receptors. Okay, so in this case, acetylcholine is released by the neuron just fine. It goes across that synaptic cleft, but it has nowhere to bind to, because basically those receptors are being blocked. So again, the muscle will never fire. People learned about this. It's now actually used in surgery to help relax muscles during surgery. Obviously, it's done in very localized fashion. Okay. Here's a picture of a black widow. It doesn't have quite your typical coloration on it, but it is in fact a black widow. Its venom works in a very different way. It causes paralysis in a sort of the polar opposite of these other two. Instead of decreasing the release of acetylcholine, it basically causes a massive dump of acetylcholine. Remember, the thing that's going to break it down in that synaptic cleft is acetylcholinesterase, right? We just mentioned that in the slide before. So that's what breaks it down, allowing the muscle to relax. Now, if you're dumping out so much that acetylcholinesterase can't degrade it, can't break it down, what you're going to end up with is a very prolonged muscle contraction, like a tetanus. So basically, you're frozen, you're paralyzed, stiff, because every muscle in your body is contracting. And not so much you, black widow toxin isn't quite, but if you're a smaller animal, that's what happens. They're paralyzed not because their muscles won't move, but they're basically fused, rigid. Okay, and that's how black widow venom works. All right. So we're already at about 20 minutes, so I'm actually going to stop this video here. We'll pick up on muscle metabolism in the next video. I know this is quite a bit of a lot, so if you have any questions, feel free to email me or ask me in class. All right, until next time, I will say goodbye.